Good afternoon, everybody. My name is Winston, and I'm an undergrad at Dr. Kimberly Gukowski's lab. It's my great honor today to introduce our keynote speaker for this afternoon, Dr. Thomas Südhof. Dr. Südhof earned both his doctoral and medical degrees from the University of Göttingen, and his doctoral thesis was completed at the Max Planck Institute for Biophysical Chemistry. For his postdoc training, Dr. Südhof joined Nobel laureates Dr. Michael Brown and Dr. Joseph Goldstein at the University of Texas Southwestern. There, he studied cholesterol metabolism and identified a sequence element of the LDL receptor, work that opened the door to genetic analyses of this receptor in patients with atherosclerosis. Upon completing his postdoc training, Dr. Südhof established his independent laboratory at UT Southwestern. Faced with the choice between returning to clinical training or pursuing research, he dedicated himself to one of the central questions in neuroscience. What is the molecular mechanism of neurotransmitter release underlying all of synaptic transmission? His group identified essential proteins at the presynaptic neuron that control the release of neurotransmitter, including synaptotagmin, which are the calcium sensors for vesicle fusion, and many, many more. These pioneering discoveries on the machinery regulating vesicle trafficking earned him the 2013 Nobel Prize in Physiology and Medicine, awarded jointly with Dr. James Rothman and Dr. Randy Sheckman. His work laid the foundation for much of our current understanding of synapses, advancing both basic cellular neurobiology and translational research in neurological disorders. Today, Dr. Südhof is the Aaron Goldstein Professor at Stanford University, holding appointments in molecular and cellular physiology, neurosurgery, with courtesy roles in neurology and psychiatry. His lab continues to investigate how synapses are formed, specified, and altered in health and disease, questions central to understanding how the brain develops, adapts, and deteriorates. Please join me in welcoming Dr. Südhof.
Good afternoon, everybody. Can you hear me? Yeah, good, wonderful. Thank you for this introduction. Makes me feel old. Really, it's, what can I say? So today, I will talk not about any of the stuff that has been mentioned in the introduction. I am really grateful to be given the opportunity today to talk to you about some work that we've been doing recently. Our focus has changed over the last decade or 15 years from understanding how synapses release neurotransmitters to understanding the molecular logic that guides the establishment of neural circuits. And by way of background, let me just remind you that neural circuits are thought generally by neuroscientists to underlie brain function, all of brain function, together with the support or the collaboration of other types of signaling in cells in the brain, but it is clearly the sort of the fundamental basis of information processing. And neural circuits process information at synapses, which are the points at which one neuron communicates with another. And synapses are the fundamental computational units of the brain. Cell biologically, synapses are intercellular junctions. They're basically like other junctions connected by adhesion molecules, but different from standard intercellular junctions. They're specialized for signaling. Synapses are very different depending on the nature of the pre- and postsynaptic neuron. Nevertheless, there's a canonical design that is shared by all synapses and that you see here schematically, which includes presynaptic vesicles that abut the synaptic junction, an active zone where the synaptic vesicles fuse to release the neurotransmitters, and postsynaptic receptors that are embedded in a postsynaptic density, a scaffold, a protein scaffold. Our key proposition that underlies the work that I'm going to talk about today is that synapses are organized by adhesion molecules, generally and specifically. In order to address this, we need to first consider that synapses are actually not simply, or synapse formation is not simply a developmental process. So very often when I talk to people, they think that in development we establish synapses and that's it. No. In development, we do establish synapses, largely guided by genetics, but then afterwards, the specifics change. And this is schematically shown here in this slide, which shows that initially during development, you have your neurogenesis, you have axon guidance, dendritic arborization, but then nascent synapses form, they mature, and they become eliminated, and this process happens over and over throughout life, resulting in a continuous rejuvenation, if you want, or replacement in a continuous plasticity that is structural, not only functional. And we believe that this is orchestrated by synaptic adhesion complexes throughout the process, both in terms of the formation as well as the elimination of synapses. A key event, a key piece of data to support this proposition was proposed in this paper here that I'm showing, published a couple of years ago, where in vivo STEP microscopy was used to measure spines as a proxy for synapses in the hippocampus during several days. And what you can see here in these images is that from day 0 to day 2 to day 4, already there's a dramatic change in these spines. And this is quantified here at the bottom to illustrate that the synapses are continuously being lost, or spines are continuously being lost and replaced by new synapses, that the number of synapses remains constant, but that the half-life of synapses in the CA1 region of the hippocampus, in vivo, in mice, is only a few days, suggesting that there's a continuous process of physiological elimination, not pathological elimination and replacement. In order to study how this is actually encoded by genes and mechanistically affected, we have at least two major challenges. One is we need to understand what the molecular mechanisms are that actually mediate all these processes. And we also need to understand how different synapses differ, how their differential properties are being encoded by distinct genetic programs. And in order to do this, we are following the proposition that all of this is based on transsynaptic signaling mediated by synaptic adhesion complexes. The word adhesion here is a bit misplaced because these complexes really are signaling complexes, they're not adhesion complexes. And we are trying to identify these complexes. We're trying to understand how they change as a function of activity, to understand better plastic plasticity in the synapses, and we're trying to shed light on synaptic dysfunction in various diseases. I'm not going to talk about any of our disease work today, I'm sorry, and I'm not going to mention the eye, sorry, okay? I'm going to try to tell you about some fundamental work that we have performed in order to better understand the most basic proposition here, which is that transsynaptic interactions, transsynaptic signaling is really what organizes synapses. And in order to talk about this, let me just tell you that many candidate synaptic adhesion molecules have actually been described, some of which are shown here in this schematic overview. There's the central molecules of presynaptic neorexins that, for example, bind to postsynaptic ligands, which we have studied extensively. And there are many others. Thus, we aim to systematically determine which of these transsynaptic signaling complexes are physiologically significant and how ensembles of transsynaptic complexes collaborate in building synaptic junctions. Now, I'm not going to talk about the systematic approach. Instead, I want to talk about one particular class of molecules because they turn out to be especially interesting, at least in our view. And these molecules are molecules that we recently discovered are key in establishing synapses in the hippocampus. And specifically, these are postsynaptic molecules called latrophilins that are adhesion GPCRs and that bind to presynaptic ligands, in particular tennurins and flirts. And so this is what I will talk about today. And so my discussion with you today will have 4 parts. I will first discuss with you an introduction to latrophilins and their complexes. I will then talk about our understanding of how latrophilins act as adhesion GPCRs in synapse formation. Subsequently, in part 3, I will switch to tennurins as heterophilic adhesion molecules that are presynaptic. And finally, I'll try to, in the shortest part of this talk, pull this a bit together and try to tell you about work where we're trying to biochemically reconstitute all of these complexes into one big complex. And so let me introduce you to latrophilins. So latrophilins are adhesion GPCRs, which means that they are GPCRs that contain extracellular adhesion domains. And in the case of latrophilins, these are lectin-like, olfactomedin-like, and hormone-binding domains that are making up the extracellular pieces together with a so-called gain domain, which we discovered more than 15 years ago, and which is shared by all adhesion GPCRs. After C-terminal to the gain domain, there's a 7-transmembrane GPCR moiety and a large cytoplasmic sequence. There are 3 latrophilins, 1, 2, and 3. I'm going to talk only about 2 and 3 today, not about 1. Latrophilins bind transsynaptically to multiple ligands, in particular tennurins and flirts. Tennurins bind to the lectin-like domain, flirts bind to the olfactomedin-like domain. Tennurins and flirts are very different types of presynaptic adhesion molecules. Crystal and cryo-EM structures enable an atomic model of the transsynaptic latrophilin-based complexes. This is put together from multiple studies, from multiple labs, including our own, which shows that there's one big complex that is formed by latrophilin in this synaptic cleft, together with tennurins and flirts. Flirts are relatively small molecules, loose-enriched repeat molecules. Tennurins are huge molecules, evolutionarily fascinating, which is another talk, and that form these complexes together with flirts, and they can bind simultaneously, as shown here in this diagram. And in addition to that, as I mentioned already earlier, latrophilins are GPCRs, and they bind to, obviously, to G proteins on the cytoplasmic side, as shown here. So this complex together is basically the latrophilin-based complex that I'm going to talk to you about today. I already mentioned that all adhesion GPCRs contain a so-called gain domain that is just outside of the transmembrane region. And this gain domain is kind of interesting because it is an autoproteolytic domain that spontaneously autocatalyzes a cleavage. But after cleavage, the 2 pieces remain attached together. And they remain attached together because the new N-terminus of the C-terminal piece remains stuck as in the rest of the domain. But they can be pulled apart, as I will discuss in a minute. So this is the fundamental structure of the complex I'm going to discuss today. And as I already mentioned, this autoproteolysis of the gain domain of all adhesion GPCRs has led to a general hypothesis of how adhesion GPCRs might function, which is that it might be occurring that mechanically the extracellular domains are just pulled off the gain domain after it's cleaved, which exposes a new N-terminus, and that this new N-terminus serves as a tethered agonist that then activates the receptor. Indeed, there's many papers claiming, for example, for latrophilin, that that's the mechanism how it works. So the idea here, in other words, is that mechanical force exposes a new N-terminus that then activates the adhesion GPCRs, and this is the most parsimonious current model for adhesion GPCR signaling. In the hippocampus, latrophilin 2 and 3 are co-expressed in the same postsynaptic CA1 region neurons, but function in distinct synapses in the same neurons. And specifically what you see here is a schematic view of the hippocampus, you can see that the CA1 region neurons receive inputs from two primary areas in addition to many other areas, namely from the Schaffer collateral pathway from the CA3 region in the stratum oriens and stratum radiatum, and from the perforin path in the stratum lacunosum moleculare. Latrophilin 3 functions exclusively in Schaffer collateral synapses, latrophilin 2 exclusively in the entorhinal cortex-derived synapses. When you knock out latrophilin 3, consequently, you only impair the Schaffer collaterals. When you knock out latrophilin 2, you only impair the entorhinal cortex synapses. First overall question here thus is, do latrophilins transduce ligand signals, and does their function involve the tethered agonism that I discussed before, that is exposed when the extracellular domains are basically pulled off? And so to address this question in studies that we performed some time ago, 6 years ago, we performed rescue experiments in the latrophilin 2 and latrophilin 3 knockouts, in which we used mutants that would specifically impair one or the other function. And specifically, one mutant abolishes flirt binding without affecting anything else. Another mutant abolishes tenurin binding. And a third mutant abolishes the tethered agonist and the autoproteolysis, both by a point mutation in the gain domain. We then used these mutants for rescue experiments. And what we found, somewhat surprisingly, is that whereas, as you can see here, using MEPSC frequency measurements as a proxy for synapse density, that the wild-type latrophilin 3 fully rescues the mutation, the effect of the mutation shown in red, but that neither the flirt binding mutant nor the tenurin binding mutant could rescue. However, the autoproteolysis mutant rescued completely, suggesting that both ligand binding sites are required, but autoproteolysis and the tethered agonists are not essential. And since then, other people in other systems have confirmed this for latrophilins, in that in no system at this point is the tethered agonist actually involved in latrophilin function. We were surprised that you needed two ligand binding sites in the same molecule for synapse formation. And so we wanted to test this in vivo, using in vivo knockout and rescue experiments as shown here in the slice physiology data, demonstrating in input-output curves, again, that when you knock out latrophilin 3, you get a loss of Schaffer collateral inputs, as shown here in this input-output. This is fully rescued with wild-type latrophilin 3, but not rescued with either one of the ligand binding site mutations, confirming that indeed the two different ligand binding sites are essential. Furthermore, to elaborate this further and to be certain about these results, we performed retrograde pseudotyped rabies virus tracing as an independent method. In our lab, we're trying to always use at least two independent approaches to try to confirm or test a conclusion. And what you see here is that in these experiments, you can label starter cells in red, shown here in the top slide, top panel, left panel. And the way this works is that at the end of the experiment, which is rather convoluted, all cells that form synapses onto the starter cells will basically become green because they express a green fluorescent protein. And what you can see is that, as you would expect in CA1, both sides of the hippocampus, CA3, synapse onto the CA1 region. In addition, as you can see on the right, a small part of the entorhinal cortex also synapses onto these starter cells in the CA1 region. You can quantify this, and what you can see on the right side here is that indeed, when you knock out latrophilin 3, you lose most inputs. You can rescue them fully with wild type, but neither ligand binding mutant rescues. Whereas the entorhinal cortex inputs that depend on latrophilin 2 are not affected, confirming the conclusion. So collectively, these results suggest that you need the latrophilin tenurin flirt complex, this huge complex that I described to you, as a key element of synaptic connectivity in the hippocampus. And this result is summarized in this overview here, where on the left you see a schematic CA1 region neuron with the inputs from different brain areas and the requirement of lateral filament 2 and 3. And on the right, you see a schematic of the complex that I showed you earlier as a structural assembly. So thus, the tenurin lateral filament fluid complex is a constituent component of many synapses that is required for the organization and synaptic transmission. And so the question then arises, how does this actually work? Having established this, we wanted to know what is the mechanistic basis. For example, do lateral filaments actually function as GPCRs? How is this coupled to the presynaptic side? How does such a complex organize synapses? And so this is what's gonna be the rest of my talk. I'm first gonna talk now in this next section on lateral filament function before I move to the presynaptic side. And so the initial question, as I already mentioned, is, do lateral filaments actually function as GPCRs? Or are they just adhesion molecules that happen to have a GPCR moiety? And so to address this question, again, we started off by making a mutant that wouldn't be able to couple to G proteins or arrestants for that matter, by inserting a T4 lysozyme moiety into the third cytoplasmic loop as shown here. And this actually does impair or abolish G protein signaling, because when you overexpress lateral filaments and hex cells, you get very high constitutive activity of GPCR signaling, as shown here with the protein kinase A activity measurements, where you can see that in the case of wild-type lateral filament overexpression, you have this massive protein kinase A activity enhancement that is induced by an increase in cyclic AMP signaling. This is blocked with a phosphodiesterase that hydrolyzes cyclic AMP, of course, and it is not present as soon as you have the T4L mutant, demonstrating that that T4L mutant is inactive in GPCR signaling. So we wonder, does this impair synapses? And it does, using a variety of approaches. I'm only going to show you one piece of data here for this conclusion. This mutant, again, abolishes rescue, suggesting that you do need the adhesion GPCR signaling mechanism. The adhesion alone is not enough. You actually have an active signaling process that is required here. And these results were confirmed by independent methods and were similarly obtained for lateral filament 2. So this leads us to this idea that ligand binding is somehow coupled to GPCR activity, and that mediates synapse formation. Our work thus suggested that lateral filament 2 and lateral filament 3, in fact, couple to G-alpha-S and cyclic AMP signaling. And here we encountered a problem in the literature because others' labs had shown, again using hex cells, that lateral filament 3 is coupled to other G-alpha proteins, but not to G-alpha-S. In fact, they showed that it's the opposite, that lateral filament 3 actually lowers cyclic AMP. It doesn't increase, but it decreases, which was somewhat the opposite to what we were saying. And so that puzzled us for a long time until more recently we discovered extensive alternative splicing of lateral filament 3 that, as I will tell you, explains this conundrum. Specifically, what we've observed is that there's massive alternative splicing in different parts of lateral filament 3, in particular at the C terminus, where there are two variants encoded by exon 31 and exon 32 that predominate, as well as a third variant that is very rare. When you look at the different expression, differential expression of these two different variants of lateral filament 3, what we found is that they're differentially expressed, as shown here, for example, in this single-cell RNA-seq data of excitatory inhibitory neurons in the brain, where exon 31 is more abundant in inhibitory neurons and exon 32 is more abundant in excitatory neurons, suggesting that this is a regulated process, that this alternative splicing is not stochastic, but regulated in some manner. And so we asked whether this might also be regulated, whether this alternative splicing might also be regulated by activity. We again went back to published data, transcriptomic data, and analyzed alternative splicing in these data. I'm not going to show you a lot of these data. I'm just going to show you one piece of data which comes from the Greenberg lab, where you can see that animals treated with kainate, which causes generalized seizures, have a rapid induction of C-Force expression that is measurable at 2 hours but subsides at 10 hours because, as is well characterized, this is an immediate early gene that you expect to go up. When you now look at exon 31 and exon 32 in these bulk RNA-seq data, so these are not single-cell RNA-seq data, you can see that exon 31 goes up and exon 32 goes down, as you would expect if exon 31 goes up. As a function of the activity, already after 2 hours, it's up, and after 10 hours, it's still up, suggesting that there's an activity-dependent long-term change in alternative splicing in the CA1 region of the brain as a function of kainate administration. So does this have any functional consequences? Initially, we thought this can't have functional consequences because this is the C-terminus way beyond the place where actually G proteins are supposed to bind. In fact, this part of the lateral 3 is not in any of the structures that have been published, and many structures of lateral 3 have been published. But when we analyzed this in collaboration with Brian Roth at the University of North Carolina, using his two-path assay as shown here, what we found is that the G protein coupling strength between these two principal variants is totally different. And specifically, what you see here is on the left that exon 31 couples to G alpha S, but exon 32 anti-couples. It's the opposite. It's basically prevents G alpha S coupling. And this is reflected on the right by the fact that only exon 31, but not exon 32, caused an increase in cyclic AMP levels in transfected HEX cells. Together, these results suggest, first of all, that there is a specific regulation of G protein coupling by the alternative splicing that is activity dependent. They also show that in addition to G alpha S, there is a more promiscuous coupling of lateral filarin 3 with other G proteins, which is consistent again with structural data that showed that there's quite a few G alpha subunits that can potentially bind to lateral filarin 3. So, what does this mean functionally? Does this mean functionally that we are having a molecule that is coupled to G alpha S and that mediates this? G alpha S that requires G alpha S coupling for synapse formation, or does this generally act as a sort of G protein coupled receptor that does all kinds of things? And so to address this question, we used an acute CRISPR knockout strategy to specifically manipulate exon 31 alternative splicing. And this is illustrated here on the left, whereby we use on top in blue just a general knockout of lateral filarin 3, and in red, a specific knockout of only exon 31. And you can see that this actually works by measurements of the protein levels. When you do a general knockout with an upstream exon, there's no lateral filarin 3 protein. When you only knock out exon 31, then the protein levels of lateral filarin 3 are not changed. They remain constant. So you can specifically delete one exon without changing the protein levels. And this is also shown in the mRNA quantifications, where the exon 31 deletion leads to the same decrease in exon 31 mRNAs as the complete knockout, whereas exon 32, shown on the right, goes up after the exon 31 knockout because, obviously, if you can't splice into exon 31, you're going to splice into exon 32, so you have a more higher abundance of exon 32. We then measured the effect of these manipulations in vivo on synapse formation using, again, retrograde pseudotyped rabies virus tracing. And what you can see here is that both the IPSI and the contralateral inputs onto the CA1 region neurons that had been manipulated are equally impaired by the global knockout of lateral filarin 3 and by the exon 31 knockout of lateral filarin 3, whereas the entorhinal cortex inputs are unchanged, as you would expect, because they depend on lateral filarin 2 and not lateral filarin 3. So together, these data suggested that exon 31 is essential. And that therefore G-alpha signaling must be playing a role in the establishment of these synapses. And this is, just having latrofilin 3 there is not enough. You actually need specific signaling pathways, as illustrated here, where GPCR activation mediates, among others, cyclic AMP induction that then is involved in synapse formation. So a general emerging hypothesis is that regulated alternative splicing amplifies the information inherent in a gene and accelerates regulatory processes because they occur at a much faster rate, as shown here, for example, in less than 2 hours, than gene transcription changes, which require generally more time, especially for large genes like lactofilins that take many hours to transcribe. Is cyclic AMP signaling the whole story? Is this how this works? You get activation of, you get basically tethering to attenuants and flirts, and then G-alpha S signaling, and that makes synapses. It is not, and that's not surprising because G-alpha S, after all, is a very general G protein that mediates a lot of signaling in the brain. Alone, it's hard to imagine that that would explain how a synapse is being organized. And as it turns out, the G protein, the C-terminus of latrofilins is known to bind to shanks, as we discovered many, many years ago. And shanks are components of the postsynaptic density. They are constitutive components of the postsynaptic density that also do other things, by the way. But within the synaptic density, they are thought to be involved in forming that scaffold that anchors the neurotransmitter receptors at excitatory synapses to the postsynaptic density. And what you see here is a schematic of how this might be working. As you can see here, the shanks are just one part of a larger assembly that also involves SYNGAP and SAPAP, PSC95, and others that all, by the way, have been deeply implicated in various diseases, in particular autism. And so the question then arises, what is the consequence of this shank binding? Is the shank binding of the latrofilins 3 actually involved in synapse formation? To address this question, we use the same specific CRISPR strategy that I mentioned earlier, but now instead of deleting all of exon 31, we only delete the C-terminal 3 amino acids using CRISPR. And what you can see here, that this again does not change the protein levels. It also has no effect on exon 31 itself, which is still fully transcribed, as you can see here, but it changes or suppresses excitatory synapse density, suggesting that you do need a shank binding site of latrofilin 3 for its function, and indicating that you have a dual pathway in that the latrofilin 3 must couple both to the G-alpha S and possibly other G proteins and to shanks and possibly other PTZ domain containing proteins in order to organize synapses. So why are the shank binding sequences of latrofilin 3 essential? As it turns out, in recent years, Mingji Zhang's group at Hong Kong, now at Shenzhen, performed some really brilliant experiments demonstrating that the PSD scaffold proteins in the postsynaptic density are forming phase-separated domains. And so we wondered whether these phase-separated domains might be related or somehow connected to the latrofilin 3 functions that we described. And so we repeated Mingji Zhang's work and then extended it towards latrofilin 3. What you see here is such an assay as imaged by microscopy. You can see in the top line a reproduction of Mingji Zhang's beautiful results demonstrating these phase-separated condensates that you have when you mix all these proteins together in the right proportions. When you add latrofilin 3, full-length latrofilin 3, 7 transmembrane regions, everything there, a recombinant purified full-length latrofilin 3 with exon 31, it incorporates into these condensates and actually forms a shell around the core, as you can see here. Exon 32 containing full-length latrofilin 3 doesn't do this. Deletion of the shank binding sequence also abolishes this recruitment. suggesting that latrophilin 3 is recruited to the condensates, or that latrophilin 3 recruits the condensates via its binding to SHANK3 and possibly other binding activities. What happens if you add latrophilin 3 ligands? Well, maybe not surprisingly, you get a clustering of the condensates. Tenurins alone and flirts alone induce mild clustering. If you add them both together, you get these massive sort of clusters of condensates together, suggesting that these multivalent ligands cause a multimerization of the latrophilin 3 and thereby cluster the condensates. And this is specific because the exon 32 containing latrophilin 3 doesn't do it. Deletion of the SHANK binding sequence abolishes the effect. So the model cartoon that emerges to explain, therefore, what we have observed is that you have this complex in the cytoplasm, in the synaptic cleft, and this complex clusters latrophilins, not shown here, and thereby causes an activation of a dual pathway, signaling pathway. One is G-alpha-S and possibly other G proteins, cyclic AMP signal and PKA, and the other one that converges is the recruitment of these postsynaptic scaffold condensates to the binding sites. And that these two convergent pathways are required for synapse organization. Having observed this, we then went on and asked, well, what happens actually on the presynaptic side, where tenurins are binding to postsynaptic latrophilins? And so the first question here was actually whether tenurins are indeed synaptic, because that had not been previously studied. And so we showed, using super resolution microscopy, D-stone, that as shown here, with a double labeling for latrophilin 3 and either bassoon for tenurin 3 and either bassoon, a presynaptic marker, or HOMA1, a postsynaptic marker, that tenurin 3, endogenous tenurin 3, is localized within the synaptic junction, and in the synaptic junction is localized to a nanocluster, to a little domain within the junction. Sometimes there's 2 or 3 of those. Most of the cases, there's only 1 of those. And this wasn't a total surprise, because we had previously found that a similar organization could be shown for neurexins, and subsequent studies have shown that other candidate synaptic adhesion molecules also form these kinds of nanoclusters, suggesting that the synaptic junction in itself is not a homogeneous structure, but is sort of forms subclusters or subdomains, which is also consistent with data from other labs demonstrating nanocolumns in the synaptic junction. So, what do we do with this? Now, there is a problem in that, again, there's lots of papers that seem to contradict what we are proposing. And specifically, these papers suggest that synapse formation involves the homophilic interaction of presynaptic tenurins with postsynaptic tenurins. And these papers are supported by structures, cryo-EM structures, such as the one I'm showing here. It was published a couple of years ago, which demonstrates these lattice of interactions. Tenurins are constitutive cis dimers, so they're always cis dimers. And here, what you can see here in this cryo-EM structure, they form this kind of lattice on the cryo-EM grid. And indeed, this is the current textbook model still for tenurins. But what we are proposing is something totally different, which is that you have a presynaptic tenurins that bind to postsynaptic latrophilins. And so the question arises, which of these two models is right? Because they are incompatible. So there's many papers that suggest this, but none of these papers has actually looked at the specific deletion of pre versus postsynaptic tenurins and the effect of such a deletion on synapse formation or synapses at all. And so we set out to analyze this by specific pre or postsynaptic deletions and then analyzed synaptic connectivity. We did the presynaptic deletion in the entorhinal cortex, the postsynaptic deletion in the CA1 region. The way this is done is shown here schematically, where you do targeted conditional deletions by stereotactic manipulations in vivo, similar to the rabies virus experiments. And when you do this, as you can see here, this is the strategy, and here you can see in the pictures on the left the presynaptic deletion, and on the right the postsynaptic deletion. You can get very specific deletions that you can actually track using co-expressed fluorescent proteins. And this allows you to ask whether the pre or the postsynaptic 10 neuron 3-4 deletion causes a change in synapse numbers. So we tested this again with multiple methods, as we always do. I'm going to show you two of those multiple methods. One is electrophysiology. Postsynaptic deletion in CA1 region has no effect on any input that we measured, including perforant path EPSCs. Presynaptic entorhinal cortex deletion has a massive effect on perforant path EPSCs. We also tested this conclusion using pseudotyped rabies virus tracing. What you can see here, the postsynaptic deletion has no effect on inputs from either the CA3 region or the entorhinal cortex. There's not even a hint of an effect, whereas the presynaptic deletion causes a selective loss of entorhinal cortex inputs because the deletion was in the entorhinal cortex and not in the CA3 region. So 10 neurons clearly function presynaptically, suggesting that the current textbook model is not correct. But we wondered if we could test this further, because there's so many papers on this, and it still is part of the general idea that we wanted to analyze this in greater detail, and we noticed that in these structures, the 10 neuron interactions are always mediated by this domain that's shown here, that is sort of different from the lateral binding site sequences. And so we wondered if we could perform deletions of 10 neurons and test their ability to rescue the synapse formation deficit that would tell us whether or not either the 10 neuron binding, the lateral binding domain, or the 10 neuron homophilic binding domain are essential for synapse formation. And so we did these experiments. I'm not going to go again into details. That was just published earlier this year. What you can see here is that when you do these experiments, again, the deletion causes a loss of synapses. Shown here is synapse density in cultured neurons. This is fully rescued with wild-type 10 neuron, and it is also fully rescued if you delete the domains that mediate homodimerization, the delta Ig beta propeller domains, as shown here. And so this suggests that indeed you don't actually need those domains that mediate homodimerization for synapse formation at all, that they're dispensable, and that these are the deleted domains. So you can perfectly get perfect good synapses without this. So this, in addition to the exclusively presynaptic function of 10 neurons, I think rule out the homodimerization mechanism as a pathway for synapse formation of 10 neurons. So then let's dig deeper. How do 10 neurons actually work? These massive molecules that are presynaptic and have this enormous domain that binds to laterophilins are always dimers and also have a large cytoplasmic sequence. And so we analyzed this further by performing various deletions and rescue experiments, and then try to understand how they might actually work. And let's move directly to in vivo pseudotyped rabies virus tracing, because they are always the most physiologically, since everything is done in vivo. And what you can see here in this summary slide with constructs that are illustrated on the top right is that the connection is from the entorhinal cortex is almost completely abolished as analyzed by rabies virus tracing with the entorhinal cortex deletion. When you rescue with a 10 neuron that lacks the cytoplasmic sequence, it can't rescue. But what you can rescue with is the one that lacks even the entire homodimerization domain, which is the EGF repeats, as long as you replace them with a short leucine zipper that also dimerizes by a completely different mechanism. And so these experiments demonstrated, in other words, that tenuins have to be dimerized presynaptically, that they have to have a cytoplasmic tail, but that they don't need to have any of the intermediate sequences except for the latrophilin binding sequence. Thus, the tenuin ICD, intercellular domain, homodimerization domain, and latrophilin binding sites are required for synaptic function, whereas the EGF-like repeats by themselves and the IG propeller domains are really not necessary. In other words, to illustrate this, you don't need any of this. All you need is the cytoplasmic sequences, a dimerization sequence, and the latrophilin binding sequence. This simplifies things enormously because it suggests that what tenuins do is that they simply multimerize the latrophilins together with the flirts in a position where the latrophilins can then function as signaling molecules to induce the postsynaptic specializations. So how can we pull all of this together? How can we think about this if we try to see this in the bigger picture? There's many different ways of how we can, in general, in biomedical research now, face the challenges of trying to understand multiple molecules at any given time. And the approach that we have chosen, as already illustrated earlier, is to try to understand this by doing biochemical reconstitutions. And so in order to do this, the first question that we had to answer then is, how are tenuins and flirts connected to the presynaptic active zone scaffolds and vesicle fusion machinery? And so we addressed this question by looking at these fusion machinery molecules that we had studied in earlier work, in fact, work that led to the Nobel Prize, in which we described that the active zone is composed primarily of a small number of essential molecules called MUNC13, RIM, RIM-BP, and a few others that are less essential. And these molecules together connect calcium channels to synaptic vesicles and promote the assembly of snare complexes whose completion and whose fusion function is then activated by synaptotagmin. And so as it turns out, this active zone scaffolding protein complex that is shown here was also demonstrated by Mingji Zhang's lab in a very nice papers to undergo phase separation and form phase-separated condensates, just like the postsynaptic site. So there seems to be, again, some kind of symmetry, different components, but on both sides, there seem to be condensates formed. And so we wondered whether tenuins and flirts might actually couple to these condensates, just like the postsynaptic latrophilins would couple to the postsynaptic condensates. I'm not going to show you the data for flirts. I'm going to focus on tenuins here. But you can see here that if you mix RIM-BP and RIM2, the two central components of the active zone that form these condensates, as shown in blue, together with the extracellular sequences of tenuins, as shown here, the tenuins do not incorporate into the condensates. However, if you add the intracellular sequences instead of the extracellular sequences of tenuins, they fully incorporate. They become part of the condensates. They become basically phase-separated together with the condensates, as shown here. And if you take full-length tenuin, recombinant tenuin, which is actually a beast, and you add them together, as you can see here, just like latrophilin with the postsynaptic sites, they basically form a shell because the cytoplasmic sequences insert, but the rest stays outside. So they form a shell around these condensates, suggesting that just like the postsynaptic latrophilin 3, the presynaptic tenuin is able to recruit the active zone condensates. So we wondered what cytoplasmic sequences bind to and recruit active zone LLPS condensates, whether there's specificity. And we used a variety of assays as always. I'm only going to show you one assay, which is the most visually, intuitively understandable, which is the recruitment of cytoplasmic sequences to the plasma membrane as a function of tenuins. And you can see here that they recruit RIM1 if it's wild-type 10-neuron-3 on top, but if it lacks the ICD, RIM1 is not recruited. And we can use this assay to analyze the effect of various mutations on the cytoplasmic sequences, and this enabled us to identify specific sequences in the ICD that would mediate the recruitment of RIM and RIM-BP to 10-neuron, membrane-bound 10-neuron. And this enabled us to ask whether this was essential for synapse formation using rescue experiments as well as condensate experiments. When we mix, as shown here, the condensates that are presynaptic condensates together with full-length wild-type RIM-BP, you get the same typical shells. When we take a point mutation that binds poorly to RIM-BP, you still get some shells, but only a few, and they are fragmented. They're no longer the wild-type condition. Moreover, when you perform rescue experiments, you can show that various mutations in the cytoplasmic sequences would impair RIM binding, also impair rescue. And I'm not going to go into this in detail because I think my time here is running out, and I don't want to go over time. So what we have then is we have all the components sort of separately. We have this postsynaptic side, we have the postsynaptic specializations, we have the presynaptic side with tenuins and flirts, and the presynaptic active zone. Can we put all of this together into a single complex that replicates a synaptic junction complex? Just mind you, this is not a synapse. This is a synaptic junction complex. Okay? That doesn't mean that this is always going to work this way. But nevertheless, I think this is a step forward towards better understanding how these synaptic junction complexes might be organized. And so we purified all these components and mixed them together in various combinations. And what you see here is that when you add full-length lateral 3, shown in yellow, to a mixture of condensates of RIM-BP and RIM as presynaptic scaffolds and postsynaptic scaffold, presynaptic are blue, postsynaptics are green, the first thing that you see is that the two different condensates don't mix. They're totally separate. They keep separate. This is important because it illustrates that the biophysical nature of condensates is not like just a hydrophobic phase. It's actually a very specific physicochemical state that depends on protein-protein interactions, and they just don't mix, as you can also see here. What you can also see is that the lateral 3 specifically only goes to the postsynaptic condensate. So this is not a non-specific interaction, it's a specific interaction, even when you have other condensates in the same solution. Conversely, if you do the same thing only with 10-neuron-3, whole full-length 10-neuron-3, you can see that it again forms a shell around the presynaptic condensates, but leaves the postsynaptic green condensates untouched. So again, specificity, again, no mixing. Now, when you have all components together, presynaptic condensates, postsynaptic condensates, full-length lateral 3, full-length 10-neuron-3, what you see here is that you now get, maybe not surprisingly, a reconstitution of junctions between the two condensates, as illustrated here, and further analyzed by stent microscopy, going sort of closer here, where you can see that the pre- and postsynaptic junctions are basically touching each other and are formed by this tenurin and lateral 3 that are labeled here in this image. This is not a full synapse. This is just a synaptic junction complex, but nevertheless, it shows you that you can actually, to some extent, replicate from purified proteins, quite a few, but nevertheless, this junction that is functionally validated in experiments that I showed you previously. So I'm getting to the end of my presentation to you. What I've tried to tell you is that the lateral and tenurin flirt complexes are an essential feature of hippocampal synapses in the CA1 region. They exhibit specificity in terms of lateral filament 2 versus 3 in collateral versus entorhinal cortex-derived synapses. And at least lateral filament 3 acts in this process by recruiting postsynaptic density condensates, whereas tenurin recruits the presynaptic active zone. And to put this into a more schematic view, what you see here is this is the complex I'm talking about. What I haven't mentioned is that there's additional components to these complexes, such as ANC5 and neurexins, that we haven't yet analyzed with respect to their role in binding to the central complex. The key message that I'd like to convey here is that there's at least reason to hypothesize that these adhesion complexes between lateral filaments and their presynaptic ligands serve to recruit and thereby enable the assembly of the pre- and postsynaptic specializations. We have only evidence for this model for lateral filament 3 that's complete. For lateral filament 2, we have partial evidence and none for lateral filament 1. To put this into the bigger picture, which is really where we would like to end up in the end, is that this is obviously not the whole story about synapse formation. Synapse formation is not going to be done by lateral filaments. It's going to be done by many molecules. Whether these molecules operate by similar mechanisms or different mechanisms, we don't know. The way how we think about this is that it is a result of convergent actions of many different signaling complexes that do different things. For example, we believe that neurexins are more specialized via their thousands of splice variants to tell synapses what properties to have. Whereas, for example, adhesion GPCRs, lateral filaments, and actually BIs as well, are more fundamental signaling molecules via the GPCR moieties and their interactions that organize the structural components. But this is ongoing work, and it will be fascinating to see if we can expand the reconstitutions that I talked about to more molecules to get a better understanding of how this might actually molecularly fit together. Let me end by acknowledging the people who have been the most important for these studies in recent years. 3 truly brilliant postdocs, among the best I've had. Chuchen Zhang, Shudong Chen, and Shuai Wang did the experiments that I described to you on the reconstitution and the rescue experiments. The initial studies on lateral filaments 2 and 3 in the hippocampus, knockouts and rescues were done by Rick Sando and Garrett Anderson, and John Yang helped tremendously. I'm grateful to my collaborators, in particular Brian Roth and Steve Quake, as well as Axel Bringer and Damit Arak. And I'm also very, very grateful, increasingly grateful, for the funding that I've received from NIMH and HHMI. Thank you very much for your attention.
Hi, Dr. Südhof. Thank you for an amazing talk. So one of the key premises of your introduction was that synapses are not permanent, and they turn over every few days. And then you showed us that in hippocampal neurons, lateral filament 3 and associated molecules are involved in the formation and plasticity of synapses. So my question is, are there differences in this machinery in different tissues? And the two extremes that I can think of is they're both sensory tissues, but one is the olfactory epithelium, where you have a continuous turnover of olfactory sensory neurons and continuous formation of new synapses. And on the other end of the spectrum is the retina, which, at least as far as I understand it, once the retina is mature, there is little to no plasticity in that tissue. So again, I wonder if there are any differences in this molecular machinery in different tissues to match the different function in those tissues.
Great question. I would love to have the answer to that question. At this point, let me step back a moment. So from our work on neurotransmitter release and the organization of synapses that we did in my previous life, I was hoping that there would be a canonical pathway that would be the same. Because neurotransmitter release, there's variants of synaptotagments, of syntaxins, of synaptobrevins, of MUNC13s, but in the end, they all work the same way. So it's very, very canonical, okay, which is great. But it turns out that in terms of the synapse formation pathways, things are going to be more complicated. And indeed, there are variations in the machinery. Latrophilin 2, for example, is only expressed on very selected brain regions and very selected synapses. I believe that you're absolutely right that variations in the machinery that I described, as well as variations in other components that I haven't had time to discuss today, for example, neurexins that are at least as central as latrophilins in terms of organizing synapses, that these variations are indeed responsible for differences in, for example, the lifetime of synapses and so on. I also want to point out that we don't actually understand how synapses are eliminated. They're eliminated all the time. When you read textbooks, people talk about microglia, but I don't think that that's the answer, because brains without microglia still have pretty good synaptic connectivity. That's pretty normal. So I think there are mechanisms that are physiologically involved and that are also are mediated by signaling. And so one way to think about this, which I'm increasingly fond of, is that, for example, alternative splicing of latrophilins that is activity dependent could also go the other way, in that a lack of activity might actually increase exon 32, which might actually decrease synapse numbers, synapse organization, and that could physiologically lead to a retraction of synapses, just as exon 31 might be synaptogenic. That's just an idea, but I think thinking down these lines, yes, there are going to be different mechanisms in different neurons, different synapses. Yeah.
Thank you.
I would like to follow what Mr. Kappa asked. Is it working? Okay, it's working. Great. Retina is stable, right? The synapses are stable.
I have no idea. Are there in vivo measurements?
That's what is believed. But during the degeneration of photoreceptor degeneration, you have massive changes in the connectivity. So the question whether this mechanism also applies during the time of remodeling could be a wonderful method to test that model for you.
Yeah, absolutely. Yeah. Yeah, so I don't know. We haven't looked at retina. I'm not aware that, in fact, there is much data except for the photoreceptor synapses, where there's beautiful studies from Martimoyanov and others suggesting really interesting mechanisms involving alphins and latrophilins. Beyond that, I don't know of any studies that have been done there, and we certainly haven't done anything.
Well, it will be time to follow your work. Thank you very much.